In the previous video, we talked about why metabolic stalls occur when NADH no longer has the ability to drop off its H+, and more importantly, the 2 electrons that it is carrying into the electron transport chain due to an absence of oxygen gas. This consequently means that there is now no way for the cell to regenerate its NAD+, and if NAD+ is absent, certain chemical reactions that depend on NAD+ to remove an H+ and electrons from a bond, both in glycolysis and Krebs cycle, will now no longer be able to take place, causing a stall of the entire ATP production process. And in this video, we're going to look at the countermeasure that every eukaryotic cell has in order to overcome this problem, which is the process of what we call anaerobic fermentation, or simply fermentation, which has multiple applications both in the human body and in human industry. The process of fermentation that pretty much everyone who has done any exercise before has experienced is what is called lactic acid fermentation, in which lactic acid obviously is produced, and this takes place when NADH, no longer being able to go to the electron transport chain, instead drops its electrons off onto pyruvate or pyruvic acid, thereby acting as an electron sink and a way to regenerate NAD+. So the way that the mechanism behind this works is that NADH normally has a positive charge on this nitrogen when it exists in the NAD+ form, and if these electrons here move and become a double bond, causing this double bond to change position in the ring here, it will cause one of these bonds with hydrogen to break off, and therefore causes the hydrogen with its electrons to add itself onto this carbon here on pyruvic acid, thereby causing one of these double bonds to break, and which can then form a bond with one of the hydrogens on water, therefore forming an OH group here, where this double bonded oxygen used to be, with the hydrogen that used to be on NADH now located here, and this chemical is lactic acid, which very frequently forms within human muscle cells when oxygen concentration is low, and this makes perfect sense, because if there isn't any oxygen present within muscle cells, that means that the electron transport chain within these muscle cells is no longer able to produce ATP. So in order to regenerate the NAD+ within the muscle cell, the NADH needs to drop its hydrogen and this pair of electrons somewhere in order to successfully regenerate NAD+, and muscle cells are where this frequently occurs because during vigorous physical exercise where more ATP is consumed than at a rate when the body is able to take in oxygen gas from the atmosphere, eventually the concentration of oxygen in muscle cells, particularly in the extremities of the body like the legs and the arms that are far away from the lungs, this oxygen is eventually going to run out. Now, most people will recognize this lactic acid fermentation because their muscles will feel incredibly sore, and if left untreated or if exercise is overdone, this is potentially very harmful for muscles, as a high concentration of H3O+, meaning a high concentration of acid, also called a low pH, is very detrimental to proteins, including that muscles are made out of, because this can lead to the extreme denaturation of muscular proteins. So managing one's exercise routine in order to prevent extreme levels of lactic acid fermentation is very important in developing an exercise schedule that works for you personally. Now, the other type of fermentation, which is also more complicated in that it actually involves two different steps is the production of ethanol. Now, this is not particularly useful in humans because ethanol is a neurotoxin as well as harmful to the human liver, but it is very important in industrial production as it is essentially this process here that is used to produce ethanol used in consumable alcoholic beverages such as beer and wine. Now, this process is a little bit more complicated in that it first involves the removal of a carbon dioxide. So here we have a decarboxylation reaction here, and we can normally recognize a decarboxylation reaction because we have a C double bond O with a negative charge here, and one of the lone pairs here forms a double bond with this carbon in the center, thereby breaking off our CO2 molecule, leaving the bond that remains to reform with a hydrogen that probably came from water, resulting in the formation of an aldehyde group here. So here, and the H plus replaces this carbon dioxide molecule and causes a compound called ethanal, not to be mistaken for ethanol. You can note the spelling difference, indicating that there is an aldehyde group here, and this molecule here is what NADH drops its electrons onto using the same mechanism that we saw with lactic acid fermentation. This lone pair on this nitrogen regenerates the positive charge to make NAD+. This double bond moves over here, thereby breaking this bond and causing the electrons and this hydrogen to attach itself to here, and again causing one of these double bonds here to break and take an H+ away from water, thereby forming an OH group here, and this hydrogen here used to be the hydrogen on NADH, but has been since removed in order to regenerate NAD+, and we can see the molecule that we have here is CH3CH2OH, also known as ethanol, the consumable alcohol that is produced during the process of alcoholic fermentation. This concludes our series of videos on the process of fermentation and what it is meant to do. If you'd like further background in fermentation, I would recommend for you to take a look at resources that deal with, firstly, lactic acid fermentation for additional background on muscular well-being, and especially if you're interested in chemical engineering, look into the process of ethanol fermentation on an industrial scale, because this reaction here forms the foundational basis for that industrial ethanol production.